Oh no, my twin sisters are marrying twin brothers, and I'm afraid that their kids will look the same. Can you all help?
Yeah, I think we can help.
Yeah, we can.
Well, I understand what your concern is because having identical twin sisters, they probably have a lot of the same genes, and identical twin brothers do as well. So, and they're about to marry each other. So their kids, you're afraid they're going to be the exact same spitting image of one another. But the good news is, congratulations, they won't be.
Oh.
So no need to worry about that. Okay, so I'll kind of explain to you, maybe all of my friends here, we can all help you out and tell you why that won't be the case. So understand that every person has different sex cells because during meiosis, your chromosomes line up randomly and are pulled apart randomly as well. This is called independent assortment. Before this step, to vary the gene pool even further, crossing over occurs in prophase. This is when two chromosomes link together and form a tetrad. In the tetrad, the two chromosomes cross over like so, and they exchange different pieces of information. So for example, this orange one has parts from the green one, and the green one has parts from the orange one. And the colors are just to make it easier to explain. Mutations could also occur. For example, the DNA polymerase could make a mistake and change the nucleotide that could change the genes of the two babies. So that will make them look different. Although the DNA is identical for both, the sets of parents, mutations, and sexual reproduction in general, and independent assortment will make them look different. I mean, you could just call it luck of the genetic draw, really. And another thing to add is, half the cells are going to be for boys and half the cells will be for girls. So it's just a 50-50 chance that they could even be completely different genders. So that's why I think they won't be the same. My friend Keegan here will help you out understand it even more. So.
Alright, this is a Punnett square. The big W stands for a showing widow's peak, and there's a small W and there's not. This is a homozygous, and this is heterozygous. And the dominant trait always shows, and no matter what, and so it'll be 100% that they'll be showing. And on this one,
welcome to the show. We're just passing through. Go on by. Sometimes friends stop by our lab in the day and they like to see what we're up to. So, go on.
yeah. This one is for lobe ears, and the big D shows it has a lobe, and the D is for attached. And these are both homozygous parents, or heterozygous parents. And there's a 75% chance that they'll have lobed ears, but there's a 25% chance that they'll have attached because there's the recessive trait pops on in there, and the D's are and the W's are the genotypes, and that's like the genetics, and then this is the physical part, phenotype.
So you're kind of saying that the genotype determines the phenotype, I guess?
Yeah.
Yeah, that's awesome. And maybe his Punnett squares will maybe be better explained when Skylar shows you his pedigrees. So maybe he can help you out some more. Don't worry, Connor. We'll keep explaining if it helps.
As you can see in Family 1 and Family 2, if he marries her, his big F comes down here, and her little f comes down here. And then if his brother marries her twin sister, his little f would come down here, and her little f would come down here. As you can tell, they each have completely different makeups. As in, this one is affected, and this one is unaffected. As you can tell in the little square, you have a 75% chance of big F, or 50% chance, 25% chance of big F, 25% chance of big F, little f, and then a 50% chance of a little f, little f. As you can tell, they're both different because one has a big F, little f, and another one has an F and an F, or little f, little f. Now, I'll leave it to Isaiah to do the rest.
Okay.
So tell him, Isaiah, what do you contribute to the piece of the puzzle?
Here's
Here's a example of codominance. The definition of codominance is a relationship between two versions of a gene, and individuals receive one version of a gene called an allele from each parent. As you can see, this is a Punnett square. It has a blood type. There's one parent's blood type. One type A and type AB. And the genome ratio would be 1 to 1 to 1, which would be AA would be 25%, AB would be 25%, AO would be 25%, and BO would be 25%. The phenol ratio would be type A is 50%, and type AB would be 50% also. We're using another example of incomplete dominance. Incomplete dominance is a genetic situation in which one allele does not completely dominate another allele and therefore results in a new phenotype. There's an example. R stands for curly, W stands for straight, and RW is wavy. Here's another example. Sex-linked traits. Big B stands for normal. Little b stands for colorblind. Sex-linked traits are only on a chromosome. They are more common in males because they only have one X chromosome.
So you're saying it's probably more likely that if the boy were to, if they were to have a boy, they would likely have one of these sex-linked traits, and if they were to have a girl, they wouldn't. So that would make them look even different too.
Yeah, it would be more different. Here's your example. Here's an example of a male karyotype. Here's your example of a female karyotype. The male has XY and the female has XX, which shows different offspring between the two. The banding pattern is different between the two because the nitrogen bases are not in the same order to code for the same proteins. Here are reasons why your niece and nephew may not be identical.
So, kind of to round it off, Connor, we kind of all gave you different examples about why they wouldn't look the same. But they may kind of look alike, but for the most part, they're going to have different traits and different personalities too. So you don't need to worry about having to try to keep them straight.
Okay, thank you.
You're welcome.